DNA polymerase is a remarkable enzyme, a true workhorse in genetics. It ensures genetic information is accurately passed on. Without it, genetic data transmission would be error-prone. This enzyme supports life, ensuring fidelity and continuity. Every organism relies on DNA polymerase to replicate DNA. It reads the original DNA strand and constructs a new one, ensuring every new cell has the same genetic makeup. The enzyme adds nucleotides to build a complete new strand. The process is a ballet of molecular interactions. Its study has profound implications for medicine and genetics. DNA replication is a fundamental process that occurs in all living cells, allowing them to divide and reproduce. At the heart of this process lies DNA polymerase, which is responsible for synthesizing new strands of DNA. As cells prepare to divide, DNA polymerase ensures that each new cell will have an accurate copy of the DNA, crucial for maintaining the organism's genetic integrity. Without this enzyme, DNA replication would be error-prone, leading to mutations that could cause disease or developmental issues. The process begins when the double helix structure of DNA unwinds, creating what is known as a replication fork. Here, DNA polymerase springs into action. It reads the existing DNA strands as templates and begins assembling new strands by adding nucleotides that are complementary to the template strand. This process is not merely a matter of adding building blocks, it requires an intricate coordination of molecular mechanisms that ensure the accuracy and efficiency of DNA synthesis. Furthermore, DNA polymerase has a proofreading ability. As it adds each nucleotide, it checks to make sure that it is the correct match for the corresponding nucleotide on the template strand. If an error is detected, the enzyme can remove the incorrect nucleotide and replace it with the right one. This proofreading function is critical for minimizing the occurrence of genetic errors, which are the root cause of many hereditary diseases. The enzyme's role extends beyond just synthesizing DNA. It also helps coordinate various other aspects of cell division. For instance, DNA polymerase interacts with other proteins that help stabilize the unwound DNA, protect the DNA during replication, and ensure that the entire process proceeds smoothly. This orchestration of activities showcases the enzyme's central role not only in DNA replication but in the broader context of cellular function and health. Within the realm of DNA polymerases, there is a diversity that ensures the effectiveness and precision of DNA replication across different organisms and cell types. Humans, for example, have several different types of DNA polymerases, each tailored for specific roles within the cell. DNA polymerase alpha, beta, and delta are the main types involved in DNA replication and repair. Each type has unique properties and functions, making them suited to particular tasks within the cell. DNA polymerase alpha initiates DNA synthesis by laying down a short RNA primer. This primer serves as a starting point for DNA polymerase delta, which then takes over to continue DNA synthesis. DNA polymerase beta is primarily involved in DNA repair, fixing any DNA damage that might have occurred due to environmental factors such as radiation or chemical exposure. This division of labor among different types of DNA polymerases ensures that DNA replication and repair are both efficient and accurate. In addition to these, there are specialized DNA polymerases that deal with extreme conditions or perform specific types of DNA repair. For example, DNA polymerase gamma is responsible for replicating mitochondrial DNA. Mitochondria, the powerhouses of the cell, have their own DNA, separate from the nuclear DNA, and require a dedicated polymerase due to their unique environment and replication needs. The diversity of DNA polymerases not only highlights the complexity of cellular life, but also illustrates the evolutionary adaptations that have allowed cells to protect their genetic material in a variety of conditions. Each polymerase has evolved to meet the specific demands of its cellular environment, ensuring that DNA replication proceeds without a hitch, regardless of the circumstances. This adaptability is key to the survival and evolution of life on Earth. Section 4. Simplifying DNA replication through analogies. Understanding DNA replication can be daunting due to its complexity. However, using simple analogies can make this intricate process more accessible. Imagine DNA polymerase as a construction worker on the site of a new building. Just as a worker lays bricks according to a blueprint, DNA polymerase constructs a new DNA strand, following the template provided by the original DNA strand. This analogy helps visualize the methodical and precise nature of DNA polymerase in action. Consider another analogy: DNA polymerase as a train running along a track. The DNA template strand is the track, guiding the DNA polymerase train as it assembles the new DNA strand, or the train cars, in the correct order. This analogy emphasizes the guided yet dynamic nature of DNA synthesis, where the path is predetermined but the process requires continuous energy and coordination. Furthermore, likening the proofreading function of DNA polymerase to an editor revising a manuscript can also be enlightening. Just as an editor scans a text for errors and makes corrections, DNA polymerase checks each newly added nucleotide and makes necessary adjustments. This ensures that the final product, whether it be a published book or a new strand of DNA, is as error-free as possible. These analogies not only simplify the understanding of DNA replication, but also illustrate the meticulous and dynamic nature of DNA polymerase. By breaking down the process into familiar tasks, such as building construction, train assembly, or manuscript editing, the essential roles of accuracy and precision in DNA polymerase activity become clear, making the science of genetics more approachable and fascinating. Section 5. Practical implications of DNA polymerase research. The study of DNA polymerase has far-reaching implications that extend beyond basic science to practical applications in medicine and biotechnology. One of the most significant applications is in the field of medical diagnostics, particularly in techniques like polymerase chain reaction, or PCR. PCR is a technology that amplifies small segments of DNA, making it possible to study genetic material in much greater detail. This technique relies heavily on DNA polymerase to replicate DNA sequences rapidly and accurately, enabling the detection of diseases, genetic disorders, and pathogens. In cancer research, understanding how DNA polymerase functions can lead to better strategies for targeting cancer cells. Some cancer treatments now involve inhibitors that specifically target DNA polymerases in rapidly dividing cancer cells, preventing them from replicating their DNA and thus inhibiting their growth. This targeted approach can potentially reduce the side effects associated with traditional chemotherapy, which affects both healthy and cancerous cells. Moreover, the role of DNA polymerase in genetic engineering and synthetic biology is indispensable. Scientists use modified versions of DNA polymerases to manipulate genetic material in the laboratory, enabling the creation of genetically modified organisms, or GMOs, synthetic biological circuits, and new therapeutic agents. These technologies could lead to breakthroughs in agriculture, industry, and medicine, illustrating the transformative potential of DNA polymerase research. The ongoing exploration of DNA polymerase also holds promise for developing new forensic techniques, improving genetic therapy methods, and even aiding in the search for extraterrestrial life by helping to identify organisms with different genetic makeup. Thus, the study of this enzyme not only enhances our understanding of biological processes, but also opens up new avenues for technological innovation and medical advancement. Section 6. Conclusion. The ongoing impact of DNA polymerase on science. In conclusion, DNA polymerase is not just a component of the cellular machinery, but a cornerstone of genetic integrity and replication. Its role in DNA synthesis ensures that life can perpetuate itself with remarkable fidelity, a process fundamental to all biological entities. The enzyme's ability to replicate DNA accurately and efficiently is vital for the health and evolution of any organism, underscoring the importance of its function in the broader biological context. The ongoing research and study of DNA polymerase continue to reveal new aspects of its mechanism and applications, impacting various fields such as medicine, biotechnology, and genetics. Each discovery not only deepens our understanding of life at a molecular level, but also propels forward the possibilities for medical and scientific breakthroughs. As we delve deeper into the intricacies of DNA polymerase, we are continually reminded of the profound complexity and elegance of life's molecular underpinnings. Furthermore, the practical applications of DNA polymerase research, from improving disease diagnostics to advancing genetic engineering, highlight the enzyme's significant role in modern science and technology. As research progresses, the potential to harness and manipulate this enzyme more effectively could lead to innovative solutions to some of the most challenging problems in medicine and biology. In essence, DNA polymerase is a testament to the intricate dance of molecules that orchestrates the continuity and diversity of life. Its study not only enriches our understanding but also enhances our ability to intervene in and guide the process of life itself, offering a glimpse into the profound impact of molecular biology on the future of humanity. As we continue to explore and understand DNA polymerase, its potential to revolutionize science and medicine remains vast and promising.